Good afternoon, everyone. Welcome to Living Labs Club workshop on soil health and socioeconomic research from the Living Lab Eastern Prairies project. My name is Dan Cox, and I'm the project manager for Manitoba Association of Watersheds. We're the lead partner for Living Labs Eastern Prairies project for the past 4 years. I would like to start by doing a brief land acknowledgement that this webinar is being hosted from Winnipeg, Manitoba, situated within Treaty 1 territory, the traditional lands of the Anishinaabe, Cree, Oji-Cree, Dene, and Dakota, and the birthplace of the Métis Nation and the heart of the Métis Nation homeland. On the agenda today in this virtual workshop, I will give a quick overview of Living Labs Eastern Prairies, a project that is near and dear to Moa's heart and been for the last 4 years, and with all of our external partners and with their collaboration with the AFC. So I'll give a brief overview of that. And then we have 3 presentations, 2 on soil health research by 2 scientists with Agriculture and Agri-Food Canada, as well as a presentation on socioeconomic research from a research scientist or senior economist, with AAFC, as well as a master's student from the University of Manitoba. And I'll be introducing them each as they come up. After each that are submitted through the Q&A function in Zoom. So please submit your questions on there, and after each presentation, we will go and cover each one as best we can. If there's any questions that we don't get to in regards to today, we will follow up with you post webinar. So what is Living Labs Eastern Prairies? For those of you that don't know, Living Labs Eastern Prairies started in 2019 and was a new approach to agricultural innovation in Canada. That brings together farmers and scientists and other collaborators to co-develop and test innovative practices and technologies to address agro-environmental issues. The 4 main pillars of Living Labs Eastern Prairies were water, soil health, biodiversity, and climate change. And socioeconomics research was integrated into all those 4 pillars of Living Labs Eastern Prairies. Overall, over 40 producers participated in Living Labs Eastern Prairies, which officially ended on March 31st, 2023. And those 40 producers covered 27 Manitoba, and there were a wide range of diversity of farm operations, small and large, crop producers, cattle ranchers. And so there was a great diversity of landowners and land that participated in Living Labs Eastern Prairies. The 4 watershed districts that participated in the program were the Assiniboine West Watershed District, the Pembina Valley Watershed District, the Red One Watershed District, and the St. Rosso Watershed District. And our other external research partners were included the Nature Conservancy of Canada and the International Institute for Sustainable Development. Just a quick map to show geographically where the Living Labs Eastern Prairies took place across Manitoba. The Upper Oak Watershed, which is in the Assiniboine West Watershed District, the Swan Lake Watershed, which was in the Pembina Valley Watershed District, the North Shannon Watershed, which is in the Red One Watershed District, and the Main Drain Watershed, which is in the St. Rosso Watershed District. Each one of these watersheds were diverse in terms of its own geography, hydrology, and farming operations, whether that be cattle or crop. There's a wide range throughout each of those watershed districts, providing a significant number of research opportunities in each one of those. All in all, those 4 watershed districts covered 5 different ecoregions of Manitoba, and was one of the reasons why they were selected in the initial process of determining where project sites would be. What were some of the activities that were undertaken under Living Labs Eastern Prairies? Regenerative agriculture, soil health and fertility monitoring protocols. Those will be spoke to today in a couple of presentations. Perennial crops and plants, water quality and water quantity management was a very popular one that'll be talked about in tomorrow's workshop. Pollinator habitat and landscape mosaic will be talked about in tomorrow's workshop as well. Spatial targeting to maximize benefits, understanding and enhancing BMP uptake, i.e. the socioeconomic research will be presented on today, as well as evaluating BMP outcomes were some of the Living Labs Eastern Prairies activities. And just quickly, some of the metrics that we were able to achieve in the first 3 years. This is not including this last year, as we're still compiling our data for the last year of Living Labs Eastern Prairies. Almost 60 presentations of results of our projects to various groups and landowners throughout Manitoba and the larger agricultural industry. We've reached over approximately 1,400 producers through knowledge transfer and outreach events, whether that's conferences, workshops like today, or any other type of farm tours and site tours. And we've been able to put out 29 different informational products, including whether that's media articles, social media, and other products produced by the researchers. So that's just a very quick overview of Living Lab Eastern Prairies. You can find more information on Living Lab Eastern Prairies at both the Manitoba Association of Watershed website, as well as the Agriculture and Agri-Food Living Laboratories Initiative website as well. So we'll get right into the presentations here at this workshop, and our first presenter is Dr. Steve Crittenden. Steve is a research scientist in soil health and nutrient management at Agriculture and Agri-Food Canada in Brandon. His research aim is to maintain or increase agricultural productivity while ensuring the long-term environmental sustainability of agricultural systems through soil health and soil fertility management. Research currently focuses on improving soil health resilience versus ecosystem trade-offs, assessing the utility of soil health indicators for corn and soybean for our nutrient stewardship for pea and soil physical quality. So please welcome Dr. Crittenden, and take it away, Steve, and you can share your presentation.
Well, thanks for the introduction, Dan. You'll let me know if you can't see or hear what's going on. Okay. I'll jump right in then. I think you introduced me pretty well, Dan. So that's who I am. Yeah. So maybe just to say what I wanted to talk about today is one example of some work we're doing on soil health at the Portage Research Center. I'll give you the details there in a second, and a little bit from what we've been doing from Living Labs as well. Both the idea being how can we relate our soil and cropping systems management to soil health and soil properties and our productivity outcomes. There we go. To do the land acknowledgement in a virtual setting, though, we're gathered on ancestral lands, Treaty 1 territory, traditional territory of the Anishinaabe, Cree, Oji-Cree, Dakota, and Dene peoples, and on the homeland of the Métis Nation. So, yeah, soil health. I feel like every presentation you see about soil health, you have to make a definition of what soil health is. So I'll do my bit there. I like to keep it simple. It's really just a way of encompassing everything that's going on in the soil, everything you can measure, every soil property. So those are the interaction, the interplay of all the biological and chemical and physical aspects of soil. But this idea of it being healthy is a little bit it's subjective. It's what we want to get out of soil. So you could define soil health as an outcome for carbon sequestration. If that's your goal, if you're sequestering carbon, it's a healthy soil. Or indeed, if your goal is to mitigate flooding, then a healthy soil might be one that keeps water on the landscape and not allow it to run off. Since our focus is agronomy to a large extent, I think our outcome has to be the primary production, so crop production. And that's what in the work that I do, I always try to link it back to primary production. And so the last bullet there, just to give a really broad sense of why we're doing these, why soil health is interesting. It's trying to design systems, agricultural systems, um, that are resilient to climate stresses. So if, and indeed in the data that I'm going to show you, if you have a drought year, you want your soil to hold water longer. And in the climate scenario where you get a heavy rainfall, you also want that same soil, that same management system to produce a soil that allows water to infiltrate. So what I'm getting at there is, with climate stresses, we need our systems to behave in different ways. And even if you take it out of that context, everyone, farmers know that each year is different, and you can get a rainy year and a wet year and then a dry year, and you may not have the same results from your management in terms of the crop production. I wanted to really briefly talk about soil structure. So soil structure is, if you look at the little brown circles there, so soil particles like clay and sand and silt, so aggregated together into aggregates, indeed, so stuck together with organic compounds for the most part. And that arrangement of those aggregates gives the soil structure. I mean, in of itself, soil structure is not important. What's important is the functions, that the interaction with our water, nutrients, and plant production there. So in the figure, I hope you can see those aggregates are arranged in such a way that there's air between, there's water films around those pores that are created by the arrangement of those aggregates, allow water to move through soil. And indeed, water can be held in those pores and within the aggregates themselves. Pardon me. And of course, there's an interaction with biology. You can see in the photo there are roots there and earthworms, soil biology interacting with those and creating those pores. So our management has an influence on soil properties and then the functions that we get from our soils. So I wanted to use an example of tillage management and how tillage influences the amount of carbon and soil structure and then therefore nutrient availability and water flow. I think it's important to, I imagine some of our producers will be in the eastern part of Manitoba, so maybe I don't need to say this, but I think broadly in Canada, I think it is important because people make an assumption about Western Canada that everything is no-till. And this is a table from Stats Canada. It's a few years old now, so I think they're redoing this now. But something like in Alberta and Saskatchewan, upwards of 3/4 of farms are doing no-till. But in Manitoba, that's about 20%. Although, more than half are doing some form of conservation or no-till. But so there's a lot of room, for, and to be clear, I think that is a result of the clay soils, the gumbo, and water management, and producers being worried about how wet things are. If I'm wrong there, I'd really appreciate you correcting me and send me a message. My email's at the end of the presentation. But so there is still a question about how to manage our tillage systems. And I know this is, going back to basics for many of you, but why exactly do we do tillage? And it is to make sure there's a good contact between the seed and the soil, so the seed can germinate. Depending on where you are in the world, you might want to incorporate manure or fertilizer, crop residues. And the photo there is one that I took from our research center in Brandon that I thought it was some sort of recreational tillage, but that was in 2022, the really wet spring, and I think they weren't able to seed anything in that field. And so the farm manager told me that they did this tillage as a way to control weeds when they couldn't. It was too late to grow a crop on it. And my sense is that there was a fair few producers around doing something similar. So anyway, so that, I mean, that's tillage right there. So that has an influence on our soil properties. And again, just to talk a little bit about tillage, probably the producers in the room are going, the plowing, no one does that here anymore. But there are just to explain the spectrum of disturbance from plowing. And you might, if it's a heavily manured soil, you may very well want to moldboard plow. And in wet regions, plowing is still done. Probably around these parts, you might be more familiar with the no-till direct seeding kind of system. But just to say that there's quite a spectrum of soil disturbance from our tillage management systems, and the way water moves, and so the nutrient availability, and so the soil health in those systems is going to be quite different. So we designed a project, an experiment at our Portage La Prairie Satellite Ag Canada Center to investigate this a number of tillage operations and their influences on soil health. And I just wanted to put this photo here to say, I don't know if you know, if you're driving through Portage on the Trans-Canada, that's a highway in the middle of the photo there, that look to the north. When you smell the sewage treatment on the south, look to the north, and that's where our experiments are. So it's a relatively clayey soil relative to where we are in Brandon. So to describe the tillage systems, our conventional system was a cultivator to 10 centimeters, sorry. And then we did a couple of maybe different tillage systems, which were meant to give a range of disturbance, soil disturbance, and moisture content. So we did a deep ripping with a rototiller to break up clumps, and we did a vertical tillage. That's a really shallow incorporation of the crop residues, and then a raised beds. I don't have a photo there of it, but that had rototilling as well, and were then sort of shaped to control moisture contents. So then into some data. I hope you can see that. Even if you can't, basically you can see the orange and the purple. That's the conventional and the vertical tillage, where there were, without the rototilling, effectively shifted the organic carbon levels within 5 years. So that's, bordering on 0.5% of total organic carbon. That's a fair shift within a short amount of time. Keep that pattern in mind with those, with a couple of treatments lower and two treatments a bit higher. These are aggregate stability data. So when I say that, I mean, so think back to that picture of the aggregates, that soil particles stuck together, creating soil structure and giving function, protecting carbon. The sort of pattern we got with a certain size fraction of those aggregates. It's the same kind of pattern where we would expect more carbon would generally, you would imagine, give better aggregation. Indeed, it's not that way across all of the fractions, but I've cherry picked one that makes sense for the story here. So in effect, in this case, it's only the blue to the purple are a statistically significant difference, but the pattern is the same there. So our management influence, our soil properties influence some of our soil functions. And so to get back to designing systems that are resilient to our extremes from climate, these are rainfall data from 2020, 21, 22. Those I've picked out because those are the years where the Manitoba pulse and soybean growers and the Manitoba corn growers funded the soil health research that we did at this site. So the total growing season rainfall is about 300 millimeters for the site. And you'll see at the bottom row there, 2020 and 2021 is about half of the amount of normal precip. So they're quite dry. I mean, really sticks out here. July 2021 had 9 millimeters of rain at the Portage site. So it was, yields were really affected by that. I think especially the canola. Whereas 2022, a wet year, or indeed, it turned out to be a relatively close to average year in terms of total precip, but that spring, that May precip level is about double what you would expect normally. So in other words, we had a couple of drought years and we had a wettish year. I know it's a busy slide, but I only really want you to look at the patterns of the colors there. So we have 3 years, and then we have the canola, corn, and soybean. Those are the crops in the rotation that we had every year there. So that pattern, I think you can see pretty well. That pattern we saw from the organic carbon and the aggregation manifested itself sometimes in the crop yields. And on that note, I think one of the challenges with soil health are because moisture, to a large extent, growing conditions are driving the plant production. Our soil health values, or how we interpret those, are not always going to be soil health better, crop yield better. It's not always going to look like that to a large extent because of the growing conditions. But so I'm going to zoom in here. This is soybeans 2021. These are just to translate the yields there. The cultivator had 59 bushels and the raised beds 47 bushels. That's a statistically significant difference. The same pattern there. So where we had drought, we had organic carbon and aggregation following a similar pattern. So where things line up in terms of growing conditions, our soil health properties line up with our yield. So I'm going to stop there on the tillage experiment, and then talk a little bit about what we've been doing with soil health in the context of living labs. So in the Swan Lake watershed, we took about 700 soil cores to get a sort of survey of soil properties across the watershed. And so that's, when I say that, you could see the vast majority, 550 were in cultivated land, but there are quite a few other, pardon me, land uses there. And so if I just focus in on the cultivated lands, I just selected a few of the soil properties, and I wanted to note for that tillage experiment, we're measuring quite a few different soil properties. And the next step is indeed we're linking those soil health properties available from commercial labs to soil microbiome data. That's the next step there. But about one of the things we wanted to do in living labs, specifically in Swan Lake, was get a sense of the amount of carbon and the amount of phosphorus across the watershed. And in terms of phosphorus, we're hoping to link with, maybe tomorrow you hear about this, they've been measuring the phosphorus in water courses, so streams, rivers, within the watershed. So hoping to link the sort of distribution across the watershed with the amount in our water courses. And I wanted to say, if you happen to be a producer that participated in this, thank you so much. It's going to be a really powerful data set. So what I wanted to comment here on the soil phosphorus stratification. This is the concentration of phosphorus soil test phosphorus with depth going along the side. And if you want to look, say, at the 15, so that's 6 inches, that's a standard soil test depth. What that little green box is telling us, that horizontal line is the median. That's the middle. So if we say that sort of below 10 ppm Olsen P, you probably need to up your phosphorus application because it's might be potentially deficient. Somewhere between 10 and 20 is the sort of sweet spot for phosphorus, and above 20 ppm we say is, they call it the agronomic threshold, where you're not going to expect a crop response to P. So what that box tells me is that the majority of those 550 sampling sites on cultivated land in Swan Lake, they're right in the sweet spot of from an agronomic standpoint. That's not to say, though, you do see, I mean, there are some points which are above. It may be that certain producers are trying to build phosphorus where it's safe to do so from a risk of loss standpoint. Or it could, there of course will be some natural variation across the landscape. And again, I know that's a busy slide. It's really just to say that what we've done at the watershed scale in terms of selected soil properties, we've also done at selected farm sites. And this is, these are, again, Olsen P by depth concentrations in 6 fields at 1 farm. And so sort of shows quite a similar distribution and stratification of P, where the P's mostly at depth because that's where we're putting it. But that in for the most part, phosphorus levels are within our agronomic thresholds. Just really quickly to comment on the organic carbon in that watershed relative to what we had at Portage. It was quite a bit higher at Portage, in some cases, more than 2% higher organic carbon at the Portage site relative to Swan Lake. So I mean, I don't necessarily know why that is. Maybe weather, cropping systems, or indeed inherent soil properties. And I'm gonna, this is the last data slide I wanted to show. This is soil pH with depth. So the red box on the left is 5 centimeters. And if you look at the green one, that's 6 inches, 15 centimeters, just to highlight the range of pHs within one watershed, where from, acidic down at 5 all the way to calcareous up there above 8. So that's a massive swing in pH within one watershed that will have an influence on plant nutrient availability. And so you might infer from that our management should reflect that variability. In other words, one size won't fit all. So just to wrap up here, that to really highlight what that soil health is, that sort of interplay of all the properties, everything going on in there, that the outcomes are subjective, what we want to get, and that our management can drive our carbon and nutrient and water cycles in these landscapes. And then to acknowledge the technical assistance we had, and in addition to Ag Canada funding, Ag Action and Pulse and Soybeans and corn growers. Thanks very much.
Thank you, Steve, very much. With some great information, some great feedback there from Living Labs work, and I really appreciate all the hard work you and your team have done over the past 4 years. And looking forward to. I know all the researchers are still analyzing a lot of results, and I'm looking forward to even further presentations of results as we go forward and into the next iteration of Living Labs as well. So really appreciate it. I'm not seeing any questions in the Q&A right now. Again, if you have any questions, please put them in the Q&A function. The panelists can answer them right there either in the box, or they can do it live. So if you have any questions, please put them in there. But now we'll move on to Dr. Taras Lychuk, who is a soil scientist at the AAFC's Brandon Research and Development Center. He holds a Bachelor of Science in Environmental Engineering and a Master of Science in Agricultural Sciences from the Ukrainian University of Nature Conservation, and a PhD in Soil Science and Watershed Management from the University of Maryland, College Park in the United States. His research at the Brandon Research and Development Center focuses on soil quality, precision agriculture, nutrient management, crop and soil models, remote sensing, and cropping system. So welcome, Dr. Lychuk, and take it away.
All right, thank you, Dan. Gonna start sharing my presentation in a second here. Can you hear me all right?
I can, yep.
Okay. All right, can you see it in a viewer-friendly mode? Okay.
You're good, yep.
All right. Good afternoon, everyone. Thank you very much for taking the time to attend this this workshop. Steve, thank you very much for your presentation. You made a good segue to what I'm gonna touch on in my presentation with regards to the research that we've been conducting within Living Labs. And even before I begin, I just wanted to say that the overarching goal of the research that we're conducting in the Living Lab is to identify factors that affect crop yield and nutrient transport in field and watershed. And we're using various tools to make that to conduct this research, which is a combination of field work with computer models that simulate biophysical, biochemical processes in the field. We also use the big data analysis, artificial neural network mechanism and tools to basically deep analyze our data to see any hidden signals that may explain the variation in the crop yield or nutrients in a particular field. I also wanted to say that this is this research is a field scale in nature. I'm going to show you later an example how we work with one particular field in the Swan Lake watershed, and how we basically make it a reality. But before I dive into our Living Lab research, I was told that we can actually present the research that is related to Living Labs, but is not necessarily Living Labs research conducted on the producer field. However, I wanted to use that opportunity to show you why in Living Labs we are doing what we are doing, because there is a reason for that from the previous research that we've been conducting over 20 years that basically drives our the avenues why we do what we do and why and how we found out about this and why it's important. Okay, so okay. So I'm gonna spend a couple of minutes, maybe 5 to 10 minutes, on one particular study that was conducted on the northwestern edge of the Canadian Canadian Prairies, which is the long-term rotation study conducted on at the research site at the Agriculture and Agri-Food Canada in Scott, which is shown here with a star. And it's basically a long-term rotation study which took place over 19 years. Yeah, this is the visual how it looked like. And the uniqueness of this study is it basically incorporated the different tillage intensities and cropping diversities into a matrix of treatments which represented the practices that are field management practices that are used across Canadian prairies. So there were three levels of input system, agricultural systems, agricultural input levels, organic, reduced, and high, and they're basically different varieties of the tillage intensities. With an organic system, there would be no chemical inputs because it's an organic production system where they would use tillage to fight the weeds. Reduced system is basically a minimum or no-till system with the reduced inputs in terms of the chemical fertilizer and pesticides and insecticides. And then the high system would be a conventional tillage with the all range of the pesticides and herbicides and unlimited, well, still within the limits, but the conventional rates of fertilizer application. There were also three levels of cropping diversity: low diversity, which was based on intensive cropping based on wheat and green manure fallow, diversified annual grains, which had a mix of wheat, barley, fall rye, and canola in the rotation, and diversified annuals and perennials, which would be in addition to the wheat, barley, they did alfalfa, mustard, and canola to this treatment level. And the reason I'm going over this is because this shows the slide here. Like, I want you to pay attention on this left graph here. This is showing the growing season precipitation across these years of the study, across 19 years. So you can see there were periods of very dry weather. There was a phase when the weather was average, and then there was for example, 2010, I think, was the highest rainfall that this place received since 1918, I believe. So it was the highest on record over the past almost 100 years. And we've done quite a bit of work with these data, and we've analyzed yield of the three crops shown here, and as well as nitrogen and phosphorus, which I'm going to show in the following slide. But you can take a closer look at these slides, and you can see that the data here is grouped based on 6-year cycles. So the first one would be the first 6 years, second one is the second 6-year cycle, and the third one is the 3rd-year cycle. And this roughly corresponds with the periods of precipitation that this research site received over the years. Like the first 6 years were on the dry side, the second 6 years were on the average, and then the last 6 years were fairly wet. And if you follow these crop yields, you can see that there is a relation between the yield and water. And like, this is not news, right? But this was quite interesting to observe in this long-term rotation study. And this connection seems obvious, but not many people have tried to incorporate this information and make a difference between, for example, with regards to the fertilizer that you input into the field, how much of the effect do you get from the fertilizer compared to moisture? For example, as I'm going to show you in the next slide, does it make a difference if you put the same rate when it's a dry year versus wet year? And the answer is yes. And it's possible in the statistical analysis for us to distinguish between these called two signals, the moisture effect and the fertilizer effect. We can also test for their interaction. And there is a third factor in addition to moisture, is the heat units, the growing degree units. We basically conducted the analysis to show that if any of these combinations between fertilizer, moisture, and the heat, which ones are more responsible for crop response, or are they equally all responsible or not? But anyways, you can see that the yield was you can follow that if you put the imaginary curve here, you can see that it quite closely follows the weather pattern. And the same also goes, this is the same graph that I've just shown you before. This one is soil nitrogen, and this one is soil phosphorus. So you can see that the soil nitrogen was lowest in the year when there was abundance of available water, which makes sense because then the plants were able to utilize that nitrogen, right? However, when you see the drier period, the nitrogen was the highest in these 3 years because at that research site, they had prescribed treatments of fertilizer application, but because the crop was not taking up that nitrogen, it kind of stayed in the field, right? And so that's an important conclusion. So it's important to account for these influences in the field management practices. Now, with regards to phosphorus, you can see that this site is gradually accumulating phosphorus, and there is an increasing trend. It's not as clearly seen with regards to the in relation to the weather as with nitrogen, but you see that the phosphorus is increasing, and I'll show you later that there are other factors that are responsible for this as opposed to nitrogen. And this, so basically, I told you this story just to tell you that we've concluded that water was one of the major yield driving factors in this particular study. Not only yield, but also the soil nitrate and soil P was affected by the tillage intensity, cropping diversity, terrain attributes, or topography, which was found to be important for soil P, soil phosphorus distribution and stratification that Steve mentioned about. That the topography would have a significant effect on how it, after phosphorus fertilizer is applied, how it then ends up in the soil profile, or if it's a subject to particulate loss with runoff, and so on, as well as cropping, growing degree days or heat units. Because if it was, for example, there were enough heat units after the seeding, there would be less soil phosphorus sitting in the ground available as subject for runoff and loss, being lost from the edge of the field. So this is just to show you that precipitation, heat units, and terrain attributes are important factors. Were found important factors in this 20-year study in northwestern Saskatchewan. Now, going to our living lab site, we found that we consider that this is an important finding from the studies that is that worth paying attention to when we plan the research at the site in a Swan Lake watershed, and this site is we work with Daryl DeVos, and I can't thank Daryl enough for his patience and allowing us to. This is the 3rd year that we've been using that field for our research purposes. So Daryl, thank you very much. But going back to the research questions, we wanted to design a system where we could also measure moisture, not only in the form of rainfall, but also deep in the soil profile, and see. We wanted to account for that in the statistical analysis to distinguish its effect as opposed to the effect of the fertilizer on crop yield. And we've done this. The way we've done it, we've basically established replicated strips with variable rates of fertilizer application at that site. And there were four levels of fertilizer application: 0%, 50% of a recommended rate, 100, and 150. 150 would be over application. And what we've done, we went to that field and we installed the soil moisture probes called Stephen Hydra moisture and temperature sensors, which would give us the measurements of electrical conductivity, volumetric water content, and soil temperature in 15-minute increments. Okay, we've broken down this field into yield management or producing zones. The green here represents high yielding zones, the yellow represents medium yielding zone, and the red represents low yielding zone. And we've basically, you can see the picture of this probe here. And in the fall of 2020, we installed several of these. We've dug the pits down to 100 centimeters, and we've installed these hydro probes across the soil profile. We connected them to the data loggers. There's a solar panel, so it's powered by the battery constantly. And we installed these stations in the high yielding zone, medium yielding zone, and low yielding zone. And we've wanted to measure the soil moisture across the years to assess its effects on crop yield and the nitrogen and phosphorus in that field. So this is a busy slide. This basically shows you the setup of our replicated strips, and then we ended up the low zone is shown here, average zone is here, and high zone is here. So there is station in each one of this zone, and this is the raw data basically across the different depths. At we have probes at 0, 50, 30. I'm sorry, 0, 5, 0, 5, 15, 30, 60, and 100 centimeters. So across the soil profile. And this is how it basically, you can see that it picks up on the rainfall. So it's very useful data to work with. These are some pictures from the time we did the installations. It was quite a bit of work, but it's this is invaluable data. It's very useful for our research. And as I said, there is a station in low zone, average zone, and high zone. There is also a fully equipped meteorological station which measures all the weather variables that there are out there: rain, solar radiation, wind, you name it. We have it for our research purposes. I did some results. Unfortunately, I don't have the numbers for 2022 for soil moisture, but I do have it for 2020 and 2021. On the y-axis here, you can see volumetric water fraction, which is basically like this one here is 32% soil moisture. In the average zone, 28%, 28.4% in high zone, and the 21.9% in low zone. So right away, you can see there is a difference in the soil moisture across the profiles in these zones, which confirms our hypothesis that moisture does have effect on the crop yield. And if it does have effect on the crop yield, it should have effect on soil nutrients as well. This one shows a small but still significant difference of just 1% between the 100% application rate of fertilizer and control check. But still, this 1% may be may make the difference in terms of crop yield from that field. Now, this one shows the soil moisture for 2021 and 2022. So you can see the lowest number in the low yielding zone, highest in the average, and average number in the high yielding zone. And for 2022, the numbers are slightly higher. So 28% in the average zone compared to 25.7, slightly higher for the bar shown in green, and this one is even the low zone is a bit higher. So these are the soil nitrogen numbers for Daryl site for 2020 and 21. You can see that the numbers were higher in 2021 compared to 2020. We have not received yet the nitrogen and phosphorus numbers for 2022 from AgWise. We're still expecting to receive them in the coming weeks, and then I will incorporate them in the graph. Same goes for soil P, slightly higher numbers. Phosphorus is quite high, as you can see, and is even higher in 2021. So just to wrap things up here, because I do want to leave a couple of some moments for questions and answers. Our next steps here are to continue to collect data, include the terrain attributes in the analysis, such as topography of different scales in terms of the assessing their effects on the yield, nitrogen and phosphorus. We're also utilizing remote sensing products in the machine learning analysis. As I mentioned, we're using the computer model to basically reproduce the biochemical, biophysical processes that occur at that particular site. So then we would be able to see from the model output to basically assess the if we can make any changes to the field management practices in terms of the, for example, the fertilizer inputs, in terms of different tillage practices, in terms of cropping rotations, and then we'll be able to see how it affects crop yield and edge of field, the nitrogen and phosphorus losses moving forward. Yes, I'd like to acknowledge these individuals listed here for their work in this project, and some references. And it was my last slide. Thank you very much for your attention.
Thank you very much, Taras. Same message I gave to Steve. Really, thank you for your time today and presentation of your data. I'm looking forward to ongoing analysis and presentation of results as you continue on with this work and analyzing results from 2022 and going forward, and hopefully there's some ongoing work there in next iteration of Living Labs as well. And so really thankful for your time today. I know you've got a hard stop at 2 o'clock. I'm seeing one. I don't know if you can see the Q&A there, Taras. There's one comment there from Mitch, but he also answered a few questions as well, but or Steve answered a few questions that he had. Any comments to either one of those?
Sorry, where do I find them?
There should be a Q&A option on the bottom, probably on the bottom of your screen, wherever your controls are.
Okay. Organic matter versus organic carbon, AES bulk density to properly determine levels. Okay, is the basic test for soil organic matter most reliably used to broadly compare results rather than detect treatment effects? For example, the PLAP soils higher OM levels are reflective of soil formation and fundamental character. So it's a question for Steve, right?
Yeah, and he answered it, yeah, in there. So
oh, he did. Okay.
Yeah, we're all good. Oh, here's one. Mitch asked you one more. Did you say that the zones are landscape position, high versus low, or productivity based?
They're productivity based. Yes. So they the way we define them is that We normalize yield across at least 5 to 7 years, and then we work with the Ag Leader SMS software. My technician does the work. He every year we receive data from producer. We clean out the outliers. We clean out a bunch of zeros, and then we overlay we add this to the stack of the previous annual yield data that we've had, and then we look at the patterns in terms of the crop yield in that particular field, and that's how we define a high, low, high, medium, or low yielding zone.
Perfect. Thanks, Ross. I really appreciate your time. I know you have another meeting at 2, so
yeah. Thank you very much.
Look forward to future presentations and going forward. Thank you, sir. Okay, we will switch gears a little bit and from the soil health side to the socioeconomic side, and we have 2 gentlemen here today to talk on this subject. One is Jun Zhao, who is a senior economist from Agriculture and Agri-Food Canada. He is involved in Living Lab Eastern Prairies, leading on the socioeconomic objectives. His current research focuses on climate change and its economic impacts on crop production and cost-benefit analysis of beneficial management practices. Joining him is Dale Penner, who is a master's student at the University of Manitoba under Dr. Von Lawley, that is exploring the agronomic and economic impacts of fall cover crops. Dale expanded his economic research on cover crops through the research affiliate program with Agriculture and Agri-Food Canada. So I'm not sure who's going first. I think it might be June going first, but whoever's first, take it away.
Yeah, Dale, can you please share the slides? Okay, thanks, Dan. And good afternoon, everyone. So today, Dale and I will do a joint presentation. So we will talk about the socioeconomic research for Living Lab Eastern Prairie. So we have 2 objectives, socioeconomic objectives for Living Lab Eastern Prairie. The first one is to look at the barriers and incentives for farmers to adopt BMPs. And the second objective is to look at on-farm costs and benefits of BMPs, such as cover crops, water retention, and tile-drainage BMPs. So the cost-benefit analysis results can help farmers make more informed decisions when considering the adoption of BMPs on their farm. The cost-benefit analysis results will also inform BMP-related policies and programs. Next slide, please. So, no, okay. No, previous slide. Okay, this one, yeah. No. First, I will talk about the main findings from the barrier incentive survey for BMP adoption in Manitoba. So this is a joint work between AFC Living Lab Eastern Prairie Socio-Economic Team and the International Institute for Sustainable Development. Marina and Bijar are my collaborators. Bijar is a policy advisor from the ISD. Marina is a policy advisor from ISD, and Bijar is a graduate student from the University of Montreal. Next slide, please. So in 2020, so AFC Living Lab Eastern Prairie Socio-Economic Team and ISD worked together to do a survey on barriers and incentives for farmers to adopt BMP. We collected information from farmers about their BMP adoption, their views on barriers and incentives to adopt BMPs, and the impact of Living Lab's participation on farmers' future BMP adoption and their social networks. So for data collection, AFC Socio-Economic Team collected data from respondents in Living Lab Eastern Prairie watersheds with help of Living Lab's coordinators and watershed managers. ISD collected data from non-Living Labs participants in other watersheds via online survey. So I'd like to take this opportunity to thank those farmers for taking time to respond to the survey. Next slide, please. Survey results, 70 farmers responded to the survey. The survey examined 11 BMPs with a focus on soil health, water management, and biodiversity. So this bar chart presents a number of respondents adopting BMPs by BMP type. Next slide, please. In terms of incentive for BMP adoption, the survey data indicates farmers are motivated by improving the environment and farm profits when adopting BMPs. The top-ranked incentives for BMP adoption are improving soil health, increasing farm profitability, being responsible environmental steward, and improving water quality and water use efficiency. Financial assistance also played an important role in farmers' decision-making for BMP adoption. Next slide, please. In terms of barriers to BMP adoption, farmers are concerned with economic and financial barriers to BMP adoption. The top-ranked barriers for BMP adoption are high upfront costs and maintenance costs, lack of financial assistance, lack of time, and uncertainty about economic benefits. Next slide, please. The impact of Living Labs participation. The survey data indicates that the majority of the respondents who have participated in Living Labs say that their participation in the program has positive impact on their future BMP adoption. The participation in Living Labs has also positive impact on farmers' social network, such as their relationships with other producers and their relationships with representatives from AFC and other organizations. Next slide, please. Based on the survey data and our analysis, we have put together a brief on socioeconomic factors affecting farmers' BMP adoption in Manitoba. The brief was posted on ISD's website. Here's a link to the brief. Next slide. So Dale, now I take it over to you, and Dale will talk about the on-farm short-term economic impacts of fall cover crops in Manitoba. Dale?
Okay, thanks, June. Yeah, so if you have questions for June, please put them in the chat and we will get to them once I've completed my portion here. So first off, my research was on fall cover crops within annual crop rotation systems. So fall cover crops are plants grown to protect the soil from erosion and to improve soil health in periods when the soil would normally be bare. So this is different than the typical cash crops, which are just grown to be harvested and sold for profit. So we're really trying to benefit the health of the soil with implementing fall cover crops into these rotations. So fall cover crops are generally planted after the harvest of the annual cash crop and grow into the winter when they're either terminated by cold temperatures or into the following spring where they could be terminated by either a tillage practice or herbicide. So with fall cover crops, we're really aiming to maximize the period of plant growth throughout the growing season. So on the left here, we have the conventional annual crop rotation, where we're seeding the cash crop in the spring and harvesting it in the fall, with that big period of time over the fall and winter months where the soil is exposed to the elements and thus subject to soil erosion. And then on the system in the right, where we're adding those fall cover crops into the annual crop rotation. So after the harvest of the cash crop, we're seeding that cover crop and we're getting that year-round soil protection of an actively growing plant later into the fall and earlier into the spring. So we can hopefully get some benefits to the soil while also reducing that potential erosion. So as part of the Living Labs Eastern Prairies, the objectives of my research were to evaluate the short-term on-farm economic costs and benefits of growing fall cover crops in southern Manitoba. So to do this, we used a partial budget analysis. So we're collecting economic data from two groups, one being the on-farm trials that were part of my master's research program through the University of Manitoba, where we're looking at field scale on-farm replicated trials. And then we also included data from the Morden Research and Development Center, where we had small plot research. And with these two groups here, we're comparing a system where we have cover crops added versus the conventional system with no cover crops. So the types of costs we were looking at, we had the direct costs, where we're looking at the additional costs of seed bed preparation, which would be land preparation costs, and the establishment costs of seeding the cover crop, and then also the seed costs. And we were also looking at the termination costs, so these would result from herbicide costs or going out and doing a tillage operation. And we also have the indirect costs or benefits. So these are the impacts on the next cash crops that is grown. So the cover crop may be positively or negatively impacting the cash crop yield. And when combined with cash crop price, we're going to see that impact on cash crop revenue. And some other costs and benefits that weren't included in this partial budget analysis would be the value of cover crops to the ecosystem services, as we're just needing further research in that area. And then also the changes in crop inputs. Due to the nature of our experiments, we weren't able to account for different crop inputs, as we're just trying to see the impacts of the cover crops themselves and not from differing levels of fertility or crop inputs. And also, we did not include any payments from cover crop incentive programs in the economic data. So with the partial budget analysis, the different sites were managed by different farmers or researchers. So we did see different seeding methods used. So we saw air drill, air seeder, and broadcast methods of seeding that cover crop. And then we also saw variable types of cover crops that were grown. So we saw a mixture of single species and multi-species mixtures. So here in this table on the right is just a list of the different types of cover crops that were grown. So we have lots of cover crop mixtures and some single species as well, with the most common being fall rye, which was grown independently at 5 sites, and then also in some mixtures in some other sites. And we also had oats that were grown in 3 sites in mixtures, and then 1 site by itself. And for termination method, we also had a big variation, as some farmers were choosing to grow cover crops that did not overwinter. So there'd be no termination costs associated with that. And then other farmers were growing fall rye or other species that would overwinter, and those required termination by either herbicide or tillage. So jumping into the results here. So we had 15 sites where we were collecting the economic data from, and these results are in 2021 dollars per hectare. So a negative cost here in this table indicates a loss or a revenue loss or a cost, whereas a positive value indicates a revenue gain or a benefit. So along the bottom here, we have the total economic impact. So we saw a mean cost of $202 per hectare. So that's a relatively large number that we're seeing there, but we did see a large range in values. So anywhere from a benefit of $64 per hectare up to a loss of $377 per hectare. Now, the majority of these costs are resulting from the impact on the subsequent cash crop revenue. So there we're seeing a mean cost of $81 per hectare, or revenue loss, sorry. And once again, we see that large range in values. So in the fields where we're seeing yield benefit, we saw a high of $112 per hectare of a gain in revenue. And in some sites where we saw a bigger loss in that cash crop yield the next year, we saw a loss of $331 per hectare. And some other major costs that came out here were the establishment costs were relatively consistent with seed costs as well. But some potential ways to limit the cost of the practice would be farmers growing farm safe seed. So whether that's bin run seed or leftover seed, is going to really limit the cost of the practice rather than going out and purchasing cover crop seed. Now, the type of cover crops that you're planting will kind of go back to your goals of the cover crop, as if you're wanting to increase the diversity of plants grown in your soil, you may have to opt for growing out and purchasing cover crop seed, whereas if you're just going for increased ground cover in the fall, maybe just using some bin run seed may be the solution there. And with termination costs, we saw a mean cost of $32 per hectare for the operation and $7 per hectare for the herbicide itself. Where we see that big low in the termination operational costs of $154 per hectare, that's coming from tillage operations. So going out and tilling your cover crop may not be the best option for both limiting your costs and also for the soil. But yeah, termination costs by herbicide were much lower than going out and tilling. So that may be an option for farmers that are growing those overwintering cover crops. And for farmers that are growing those winter kill species, there's no termination costs associated with those. So that's another way you can kind of minimize the cost of the practice. And some potential benefits. So we didn't really include these in the partial budget analysis, but there's some need for future research in these areas. So there's the potential for decreased crop inputs with the use of cover crops, whether that's resulting from weed suppression or those potential long-term nitrogen credits that we may see. Further research is really needed in those areas. And there's also additional revenue source possible for farmers growing those fall cover crops if they're going to go out and graze those fall cover crops. And another potential revenue source would also be from cover crop payment programs or from carbon credits that would come out in the future. So in the long term, for the widespread adoption of cover crops, we're really going to need to see minimizing the cost of the practice and also a balance between the benefits to the soil and also with the cost of the practice. Thank you everyone for listening. I'm glad to open it up to questions here.
Thank you very much, Dale and June. I'm not seeing any questions in there just yet. If anybody does have any questions for June and Dale, please put them in there and we'll get to them. One other thing, another item in regards to socioeconomics and cost benefit analysis, and I'll just share my screen, is a product that the International Institute for Sustainable Development put out late last year, and Marina presented on it at our conference as well, was the value of water retention beneficial management practices to farmers and landowners in the St. Rosa watershed district, and all the product, all the water retention products that are being built in that specific watershed, and the cost benefit analysis of that. So you can find this report on ISD's website. So please go there and download and have a read. Like I said, she presented it at our conference, our mock conference back in December of 2022. But please go and check it out. Any other questions? I'm going to give it a few minutes here if anybody has any questions for June or Dale on the cover cropping. Same thing, same message I gave to Ross and Steve in terms of thanking you for your time today and the presentations, as well as the ongoing research that you do and analyzing the data going forward. I don't know, John, you're a panelist, so you can speak if you like. I know you said you had a question as well, or you can put it in the Q&A.
I guess I'll just speak here, maybe.
Go for it.
Just a couple quick questions. Maybe one going back to Steve Crittenden and one for June. So maybe going back to Steve Crittenden, if he's still here.
Yes. Yeah.
What would you recommend for a producer if they want to monitor their soil health?
Hi, John. I mean, like everything that's muddy about soil health, I mean, what do you mean? Do you mean if a producer had to select one thing to measure, what would they measure? Or do you mean sort of as a system, how should?
I guess I would like to say one thing, but I don't think there's a right answer there. So maybe as a system.
I'm going to answer the first one, even though you said I should answer the second one. I mean, one of the things that is coming out of the work we've been doing with the Pulse and Soybean and Corn Growers Association is that, I mean, there's the oldies are still goodies, if I can say it like that, that there's still a lot of value in a nitrate test and a pH test, your Olsen P. That's one message that I think I'm going to give from that work, is that the value in terms of, let's say, more traditional soil tests, or what you would conventionally see on your soil test, there's a lot of value there for predicting crop performance, at least as much as, say, newer indicators have value. But if we also wanted to, like anything, like I said in the presentation, it depends on your goal. It's subjective. So you could soil test for your fertility planning for the spring, but you can also soil test to benchmark your soil properties with time. If you're going to do, you know, you're going to try something different on that field, or a different tillage system or cropping system, whatever, you can take a number of soil samples and get that data and benchmark it, and then and track it through time. Those are sort of two valuable points there, I think. Does that answer your question?
Yeah, it did. You know, there's a lot of talk out there on how to measure soil health, and I think it's important to get a good view on that. So, thank you. And I guess the question I have for the socioeconomic side is basically, what was the most important fact or point that you got out of your research?
Yeah, I think the one thing is for the socioeconomic research is barrier and incentives. So we have shared the information and the survey result with policymakers and program officials, and provide them with a better understanding of practical farm level considerations for BMP adoption, and inform BMP related policies and programs. This is one thing. And the other thing is kind of based on the survey results, there's some concerns, there's some barriers, for example, the lack of easy access to information and uncertainty about the economic benefits and environmental benefits of some BMPs. So the next step is kind of, so we will put together a fact sheet, for example, based on our cost-benefit analysis results, such as cost-benefit results of cover crops, water retention that John just mentioned, in collaboration with ISD, and also heterogeneous BMPs, and also provide input to the vendor for the Living Lab Eastern Prairies that will be shared with producers.
It's good to, it's always good to hear when information is making it to the hands of those who make decisions about programs. So that's great to hear. Thanks.
Did you want Dale to answer that one as well, John, in terms of his research?
You know, if he feels up to it, sure.
Oh. Yes, so my research was pretty broad. We just kind of touched on the economic part of it today. But yeah, we were really just, there was not that available economic data in the prairies before our research really started. So I guess providing that baseline of what producers can expect, I guess, with regards to cost of the practice, I think is a crucial step in promoting adoption and helping our producers understand the cost of the practice, and I guess potential ways they can minimize their cost of the practice as well. As far as the agronomic data, I mean, we're still in the initial years of adoption, so it may be a couple of years before we see some significant impacts on soil health or other soil properties. But even just within the first 3 years of adoption on our on-farm trials, we were seeing those improved soil health scores, but which is surprising in such a short period of time, but it's also promising for the adoption of cover crops in Manitoba.
Well, thank you. That's all my questions.
Thanks, John. Anybody else have any other questions? And please put them in the Q&A. We'll give it a few more minutes here. Unfortunately, we had one presenter unable to make it today due to illness, so we're going to be a little short on presentation today. But you know, it's a beautiful day outside, so you'll be able to get out and enjoy it. But if there's no other questions in the Q&A, there was one question in the Q&A that was for Taras, and unfortunately he had to run to another event. But we'll have him follow up on that question directly after we're done. Tomorrow's workshop, I hope you're all registered for tomorrow's workshop on biodiversity and water and quality and quantity research through Living Labs Eastern Prairies. We have a full agenda of presenters tomorrow, covering a wide range of topics, like I said, through biodiversity and on water retention. We do have a watershed district representative presenting tomorrow, as well as a number of AFC researchers. So please join tomorrow, same time, 1 o'clock, for a full agenda of presentations and Q&A. And so thank you to Steve, thank you to Taras, thank you to June and Dale and John for your questions at the end and there to fill some time. Really appreciate it. And we will end a little bit early today, but I look forward to seeing you all tomorrow. Thank you very much.